Hello and welcome to the Next in the JCI series, Conversations with Giants in Medicine. I'm Ushma Neal. The simple fact that you can understand what I'm saying and that you might remember any of this interview tomorrow is thanks to the simultaneous and precise coordination of communication between the billions of neurons in your brain and peripheral nervous system. My conversation today is with Tom Sudoff of Stanford University, who is at the center of unlocking the secrets of neurotransmission. His work over the last 30 years has elucidated much of what we know about the molecular mechanisms of neurotransmission and synaptic signaling. For this body of work, Sudoff has recently shared in the 2013 Lasker Basic Medical Research Prize. Thank you so much for joining us today. I'm delighted to hear a little bit about your work and your life. Do you think you could start at the beginning with a little bit about your upbringing?
So, as you can gather from my accent, I was born and grew up in Germany. I was actually born in Göttingen, which is a small university city, just very close to Hanover. My parents were both physicians, and my father was at the university. He was interested, in fact, in how diseases develop in terms of metabolism, so he was interested in particular in lipid metabolism, which is kind of ironic given my later career. I went through the regular schooling in Germany, and while I was a teenager, my father died, and so that certainly influenced my development considerably. After school, I decided to study medicine and to become a physician myself, not because of the family tradition, there was none apart from my own parents, but because I felt that medicine was the best way to learn as much as possible about the world and to be in a later position to do something practical and useful. And so in Germany, the system is very different from the United States in that medical school starts immediately after high school. High school is longer. So when I started medicine, I had gone through a longer curriculum of high school, and I then started in medicine, which is a 6-year curriculum. And that's basically how I embarked on a biomedical career.
As a child, were you fascinated by science at all, or nature?
To be honest, no. You know, it's what always fascinated me and still fascinates me is knowledge, but insights, understanding. What also fascinates me, even though I've been maybe not very good at it, is sort of what happens to humanity in a broad sense. What I mean with that is events, from natural disasters over positive developments such as technology developments that enable progress. For example, there's so many it's almost a surplus of riches, but for example, the development of agriculture 4,000 years ago, the development of the steam engine 200 years ago, the development of computers 50 years ago. So when I was brought up, I had a very broad background that included a lot of humanistic types of learning, including classical music and literature, visual arts, as well as a broad background in natural sciences. And I wasn't interested in science as such, and I didn't become interested in science as such until I was in medical school, and I realized that our ways of approaching disease, of trying to help people who are sick, of trying to be of some help to anybody, depends on an understanding of the fundamental facts. And that's really when I became interested in science.
Now, in terms of medical school, did you finish in what in America we would call a traditional internship and residency in your clinical training?
So my career path is a little unusual. I finished medical school, I went through an internship, And afterwards, I actually had intended to pursue an academic career in Germany. And I was particularly interested in clinical medicine. And an academic career in Germany in clinical medicine involves scientific training, requires scientific training even. And I decided to obtain that scientific training beyond my doctoral degree by coming to the States. And I did this with the full desire to go back and complete a full residency and become a real doctor. And even after I had finished my postdoctoral training with Brown and Goldstein, I still had thoughts of going back to residency, and it was only pretty much after a lot of thinking that I decided not to, and I still somewhat regret that, but it's, I suppose, irreversible by now.
So let's go back to your doctoral work, or your early interest in actually doing lab research, which, as I recall from your biography, you did working on chromaffin cells with Victor Whitaker.
Yeah, so this is also a bit unusual because at that time in Germany, medical school meant basically that you were supposed to fulfill certain requirements, both theoretical and practical, but there was a lot of flexibility and freedom. And when I went through the preclinical sciences, which takes in Germany for 2 years where you do nothing but just study basic biology, I became interested in science. That's really when I started to think about how cells work, what do we actually know about how an organism works. And so after my preclinical time, when I started to go into the more clinical part of medical school, which takes 4 years in Germany total, I also started to work in a laboratory by the side, sort of in the free time and in the summers. And at that time, I looked for a laboratory I could work in Göttingen, where I was. And Göttingen had a very nice Max Planck Institute, which was on the mountain above the city and was founded by Manfred Eigen, a Nobel Prize winning chemist. And so I went there and tried to figure out where I might be welcome as a novice to just basically work. And Victor Whitaker, who was a director at the institute, said, well, you can work here, you can work with this assistant I have, this sort of more senior scientist who is a permanent member of my staff. And so I started working with this person. But I mean, this is something I would never, ever conceive of nowadays, and certainly would find even scandalous nowadays. But then things were pretty flexible, and I sort of worked there, often nights, and then the person I was supposed to work with left, and after half a year or so. And so I was pretty much left to my own devices with a lot of advice from Victor Whitaker, but no real sort of mentor who would be there on a daily basis. And that freedom was actually extremely inspiring in some ways, because it allowed me to try all kinds of things. And I decided to work on chromaffin cells, and actually chromaffin granules, which are the secretory vesicles of these cells, because I had learned from others in the institute that that's a source where you can purify an organelle and study the organelle. And I thought that we don't know anything about the molecular and biophysical properties of organelles, and so we might as well study that organelle. And so that's in a long-winded explanation how all this developed.
So after that experience with that mentor, how did you land in the Brown and Goldstein lab, what must have been one of the most famous laboratories in the United States?
So when I finished my doctoral thesis and finished my work in Göttingen, And as I was going through my internship, I decided that I wanted to be an academic clinician, as I mentioned before, right? And I decided that in order to be an academic clinician and to get scientific training, I needed to get the best possible training in what seemed to me to be the most interesting advances or fields at that time. And others had made me aware of Mike Brown and Joe Goldstein's work and pointed out to me that these scientists and clinicians were doing extraordinary work. At that point, they were not as famous as they are now, as they've been for the last 20 or 30 years, I suppose. They were in Dallas, Texas, which wasn't exactly, let's say, famous center of science to the best of anybody's knowledge. And so it wasn't necessarily an obvious choice. In fact, I myself was somewhat hesitant because of the location. But I went to Dallas to visit and interview, and like I think everybody in the world was incredibly impressed, and that's when I decided to apply, and I was extremely happy, obviously, that I was invited to go, and that's how it happened. Yeah.
And because they were working on cholesterol metabolism, you fell into a project wherein you cloned the LDL receptor gene.
So then historically, I arrived in '83, in the October '83, and that was a time when it was a very, very exciting time in science, because molecular biology had basically been invented in the previous decade, and it was achieving the point of maturity that was possible to clone genes and to understand how they might work. And so I was taught in Brown and Goldstein's lab basically how to do this, and I cloned the LDL receptor gene. I became completely immersed in cholesterol metabolism and receptor biology and cholesterol metabolism, because the whole field at that point of the LDL receptor and LDL and cholesterol revolved around not only the metabolic pathways that cholesterol is involved in, but equally around the cell biology of protein transport. And what is nowadays textbook knowledge and very well established then was completely unknown, which is the whole notion that you have cell surface receptors that recycle via the surface and that are regulated and that bind things and take them up, and that this is a vital component of every cell's existence, every cell's life. And so that part was a tremendously exciting experience, and I learned a tremendous amount in my 3 years in Mike and Joe's lab. In this time, I actually worked on 2 related projects, which is not only the cloning of the LDL receptor gene, but also something that had probably even more long-term repercussions, which is the identification of sequence elements in the LDL receptor gene that mediate the regulation of the expression of the LDL receptor gene by cholesterol itself. So there's if you want, some kind of feedback regulation, so that that the gene is expressed whenever it's needed. And this whole mechanism of how cholesterol regulates gene expression then led in further studies by many really wonderful postdoctoral fellows in Mike and Joe's lab to the discovery of the whole cholesterol-dependent regulatory transcription pathway, the SRBP and so on, right?
So given the excitement surrounding SREBPs and LDL, when you first decided to establish your lab, why then move on to synaptic biology? And then also as a corollary, why did you stay in Dallas?
No, there's many answers to this question. So first of all, just as a kind of career advice, okay, since I don't know how many young scientists will ever look at this, but anyway, nowadays, virtually nobody dares to change fields, especially from a postdoc to independence. Even from a graduate student to a postdoc, most people just stick to their fields because, justifiably so, people are afraid of not having enough funding. But in biology, I think there is a big danger of becoming specialists in one little area which stifles creativity, I think, and really is not necessarily a good long-term career move because you never learn anything new. You only learn new techniques, but you don't actually learn new concepts. And I still think that even though techniques are often rewarded more prominently than conceptual advances, it is the conceptual advances which make science worth doing, not the technical progress that we have all had the luck to go with. Yeah. So that's that part. Okay. So coming to the issue of why change fields. There's two reasons. One is that if you work next to true giants in science like Mike and Joe, not giants like me in this interview, but really fundamentally innovative and creative scientists, and you become independent, it is going to be, I thought, very difficult to have any real, completely independent ideas, thoughts, developments, if you are right next to them and work on the same field. So my decision to change fields was a conscious decision in part because I felt that if I'm going to be independent, I want to do something that nobody else has done before. I don't want to sort of continue what I was doing, and maybe I would have made major contributions, maybe not, but I would have always thought that many of the ideas may have been, if not consciously, then subconsciously sort of diffused. And I wanted to have my own intellectual area of.
Then why choose synaptic neurotransmission?
So the other, why choose synaptic neurotransmission? Because if you think about the brain, the most important thing in the brain, and I still think this is true despite the fact that that is absolutely debatable. But the most important event that happens in the brain is synaptic transmission. And the reason is that any communication between neurons, any information processing happens at synapses. Neural networks are formed by synapses. Synapses not only transmit information, they're also computed because they change it. They have an individual memory. They change as a function of use. They are the most fundamental information processing unit, not only information relay stations in the brain. And when I started, nothing was known about synapses except that they communicated via the release of a chemical transmitter that then was recognized by postsynaptic receptors. And so I decided to start working on this problem in molecular terms because not a single molecule had been cloned prior to this, not a single protein had been identified in molecular terms. And it seemed to me that understanding how this works would have tremendous effects on our understanding on how the brain works.
Your work over the years has focused largely on the presynaptic synapse. Why the pre side and not the post side?
Yeah, that is a very, very good question. We initially started on the presynaptic side because it was initially more amenable to a molecular dissection. When we started by purifying synaptic vesicles, often in collaboration with my long-term collaborator Reinhard Jahn, and then trying to understand their components. That was possible because of prior work which established how synaptic vesicles can be purified. Postsynaptically, not as much was known. However, during the time of this work, everything changed in the sense that postsynaptic site became much more easy to analyze due to the advance of electrophysiological technology, and because you can sort of detect postsynaptic events much better than presynaptic events. I started initially presynaptic because that was, at that point, technically the better avenue. I still work on presynaptic mechanisms because, after all, neurotransmitter is what initiates synaptic transmission and is an absolutely essential part of synaptic transmission. But our work in recent years has actually also captured many questions on the postsynaptic side, almost by necessity, because the pre- and postsynaptic side are so closely coupled. Many of the proteins that we have identified and studied have either correlates on the postsynaptic side or interact with other proteins on the postsynaptic side.
As you established your own lab, you became an early HHMI investigator, and the support of your lab has continued to the present. How did that early investment in your science shape the amount of freedom that you had to study different things, and how has it supported you through the current day?
So I consider becoming an early HHMI investigator one of the biggest pieces of luck, and I've had a lot of luck in my life, I have to say, enormous amounts of luck. But it was certainly one of the biggest pieces of luck I've ever experienced, because it gave me the ability, the freedom to just do things for many years without having to rely on the need for continuous renewal of funding. However, having said this, let me also sort of point out historically that that was a very, very different time. At that time, NIH wasn't in the shape in which it is now. It was actually reasonable for people to expect that if they wrote a good proposal, they would get funded. It was reasonable that good science would get funded. I think that that's not the case anymore. I also think that even then, HHMI was not necessarily without checks and balances. So when I became an HHMI investigator, I became an HHMI investigator together with several others. And of those others, several actually were discontinued after only 2 years. So it wasn't necessarily a free ride. It was really, from the very beginning, a culture that was very much accountable, to put it this way. And nevertheless, I think without HHMI funding, I would not have been able to make the types of observations that I made in my career. And I think it's terrific.
So synaptobrevin, synaptotagmin, RIM, MUNC18, more recently Neurexin and Neuroligin. You've spent your career showing us exactly what the mechanism of neurotransmission is and the proteins that are in there. How have you managed to have this singular focus? And can you tell us about what it was like during that phase of your career, of these discoveries?
I think it is often confusing for people who are not in this field to have, it's almost like a zoo of molecules that are all sort of mixing and matching. They all have these strange names, just like animals, I suppose. Yeah. So my focus on synapses is driven by my continued belief that understanding synaptic transmission is absolutely crucial for understanding any brain function. And high level, low level, memory, immediate reactions, anything, vision, perception, thought, consciousness. And I think we need to understand this at a level that cannot dispense with the insights into molecular mechanisms, because I would argue that molecular mechanisms underlie, for example, memory. Having said this, which is not exactly the question you asked, it does get there. So, there clearly is, I mean, the synapse is obviously going to be complicated. It's not going to be any less complicated than a ribosome, than protein translation. Right? Anything that, when you look close, becomes complicated, because if you're a physicist and you study atoms, it becomes complicated, right? So, how can we actually deal with the complexity and the many components? And one way to do this is to try to identify what components are there. Then subsequently try to figure out which ones of these many components are the ones that actually matter, and which ones are the ones that are not going to provide major insights, which ones are the ones that are going to have really consequences for our understanding, and then focus on these components. Among the list of proteins you mentioned, all of these turned out to be proteins that are of absolutely central importance. But there's many, many others I have studied over the years that you didn't mention. That I spent many, many, many years and a lot of papers on. And that to this date don't really have a well-known function, and probably are important in some way, but are not central players. I often would see biology to operate in analogy to a play, where you have central characters that execute the actual action that is displayed, and then you have peripheral people that come in and out of the play because they complement in some way. Sometimes they're necessary for understanding, but they are not the ones that actually drive the action. They're not the ones that drive the story. And in biology, if you want, it's the genes or proteins that are the central players, or the peripheral ones. And the strategy that we have pursued over the decades is to try to identify the central players. This was the 90s, the beginning of the 90s in particularly. To try to identify which ones of these central players are actually really central and which ones are more peripheral. And then to try to figure out how the central players actually work and what they do, and to try to, in particular, relate what they do to specific abilities of a synapse in terms of function. For example, the ability of a synapse to change, or the ability of a synapse to be fast, or the ability of a synapse to be regulated by other signals.
Now, in looking at your CV and reading about your work, many of your findings of the proteins specifically that I was asking you about were made around the same time as others. And the understanding of the biology of the synapse happened not necessarily only from your group, but also that of Richard Scheller, some input from Jim Rothman and Randy Schekman into the snare complex. Was it a competitive landscape or a collaborative landscape?
It was, and still is, I think, a pretty competitive landscape. But the approaches were slightly different. Richard Scheller and I pursued quite similar approaches, but our proteins rarely overlapped. There was some overlap, but it was limited. Where the most competition came in fact, was probably in the discovery of how Clostridial neurotoxins, tetanus toxin, botulinum toxins act, which was really a seminal part in that time. I don't think there was any competition in terms of the active zone, as far as I can remember. I mean, you know, RIM, MUNC13 and so on, which has, in terms of understanding synapses, become really crucial. And I don't think there was any competition in terms of synaptic cell adhesion. There is now a tremendous, very competitive field, but it wasn't when I started this 20 years ago.
Speaking of your science and your publications, in looking through your CV, most of your articles have tended to have fairly short author lists, even in this growing age where author lists can reach into the hundreds.
Yeah.
Is this part of your approach to mentoring, to work with smaller groups of scientists on focused ideas?
No, I wish I could say, yes, that this is the ideal or whatever. I think that this is a complete reflection of the approach. So I think team science is terrific because it enables doing things in a systematic manner that would otherwise not be possible. For example, genomics, huge expression studies, probably connectomics, that is understanding sort of the anatomical basis of neural circuits. Those kinds of approaches require large numbers of people working together, and the outcomes of these approaches are of really important values that will be accessible and so on forever, presumably. However, my science, my interests is really to understand how synapses work, and that at this point I don't see a team approach to that question. I think we need to really ask specific questions. How exactly what is the observation, what is the property, what are we trying to answer, and how can we answer that? And it really needs, there is no sort of industrial scale way of approaching that at this point, at least.
You've previously extolled the virtues of solid, basic, descriptive science, mechanistic science. I'm here representing a translational medical journal, and I've actually seen in looking through your CV that you've looked more at problems involving alpha-synuclein and in vivo models of autism. Are you evolving in a direction towards more in vivo biology? Are you taking on more students with the thought of going in that direction, or are you still the major proponent of basic descriptive mechanistic science?
That's a very philosophical question. I always like to hold up the example of my mentors, Mike Brown and Joe Goldstein, in this respect. I am extolling mechanistic science not only, but also for its own sake, because I believe in the value of knowledge for humanity, period. I think that knowledge is a value in itself. It's not a luxury, it's a necessity. However, on top of that, I think that any advance in treating the major neurological disorders that currently afflict the world, not just the US population, from schizophrenia and autism to Alzheimer's and Parkinson's. There's many. We don't understand any of these disorders, actually. In order to understand them, we have to have a mechanistic understanding of the processes that are disturbed in these disorders. And that mechanistic description, if you want to call it that, requires fundamental biology. And so I think that my criticism would be not that people try to emphasize translational research. I'm all for it. I would love to discover something useful. But my criticism is that you can't jump over, just basically jump into translation if there's nothing to translate. If you don't understand normal thought it's going to be impossible to understand abnormal thought in schizophrenia. I mean, you can't symptomatically treat it, sure, by serendipitous discoveries, as all schizophrenic drugs are used now, which were basically discovered by accident. But you can't actually ever get at the root of the problem. The same thing for autism, same thing for neurodegeneration. And a huge advance in these fields, for sure, was made by genomics and by what I think is a remarkable revolution of genetic understanding. But what this gives us is a bunch of genes. And what we need now is an actual understanding why these genes might be related to the diseases. That's where any progress will come from, not from jumping to trying to find drugs that treat these genes, because we don't actually know why these genes are connected to the disorders. And coming back to Brown and Goldstein, knowing that cholesterol is the culprit alone would never have led to the development of statins. In fact, it was known by the time they studied cholesterol biosynthesis and its regulation. And it was already known even how cholesterol is synthesized. The discovery of statins as therapeutic agents, which arguably is based on Mike Brown's and Joe Goldstein's work, was really only possible because they had done the basic biology. It wasn't done because they studied, they were trying to treat hypercholesterolemia. And so I think that still serves as a fitting example why, for my belief, why it's important to have a fundamental insight into the biological processes underlying a disease, not just try to go ahead and do translation.
I asked you a question about mentoring before, and I want to bring it back to that. What's your approach to mentoring the trainees that come through your lab?
My approach is to work with each one as closely as I can, to talk to them on a one-to-one basis as often as I can, and in addition to have as much of a team atmosphere as possible. So I like to have regular, frequent meetings with groups of people who don't exactly work on the same thing, but work on similar related questions, and stimulate discussions between them, even if they don't actually work on the same thing, in order for them to complement each other and to help each other. So I think that in science, nobody operates in an island alone. There's very few geniuses left. Certainly, I'm far from any such existence. And so I benefit from others, and I want to mentor people to learn how they can benefit from others, because I think that that's the best help we can get always, is from the opinion of other people.
Team science.
Yes, but team science there's different types of team science. There's team science where basically 100 people do the same thing, but each one covers a little different segment of the same thing. Absolutely essential, maybe completely boring, but crucial for the advance of science. There's another team science where people work on a similar question, but approach it with individual angles and have different complementary questions. That type of team science, I think, is just as important, and is sort of, both types of team science exist and are crucial, but they are very different types of team science. Yeah.
If you had to do it all over again, would you have picked the same path?
If I were doing it now, I often feel that the culture I grew up in Germany, was a culture which put an enormous value on the intrinsic importance of knowledge and truth. And I feel that cultural attribute has sort of become less over the last decades. And I think that nowadays I would be more interested in sort of a more, not that I would be able to do it, but in a more advocacy role, because I feel that although there's much we don't know, that we also must discuss, understand, promote, possibly, or maybe dispense with this idea that knowledge is intrinsically important and how we deal with it in a society.
So sort of like a science pundit or a knowledge pundit.
Well, in some way, things like you do. I mean, this is very, very important. And I think that we, in general, as a society, as a culture, global culture by now, there has been much less, there has been sort of a loss of consensus of what the standards are. And that leads to new questions, and I find this very interesting, and I would love to have an active role in addressing those questions.
Thank you so much for joining me today and allowing me to bring your story to a bigger audience. Thank you.
Thank you.